Good afternoon, everyone. I'm Wei, the Senior Product Manager of Novogene Europe. First of all, I'd like to thank the Festival of Genomics to invite me to join this webinar. In this talk, I will go through the Novogene transcriptomic and epigenomic solutions for human research. After the Human Genome Project, our scientific world has been transformed significantly. It changed the way we think about scientific questions and answer those stories. Good afternoon, everyone. I'm Wei, the Senior Product Manager of Novogene Europe. First of all, I'd like to thank Festival of Genomics to invite me to join this webinar. In this talk, I will introduce you the Novogene transcriptomic and epigenomic solutions for human research. After Human Genome Project, the scientific world has been changed significantly. It transformed the way we answer scientific questions. Currently, we use different strategies to sequence different omics. To meet that diverse request for the human research, Novogene provides DNA sequencing, RNA sequencing, and epigenome sequencing, as listed in this slide. We also provide primate libraries and customized analysis. While in this talk, we will focus on those two topics. First, let's go through the transcriptome services. As shown on the left, here is our workflow for each Novogene RNA project. We usually recommend our clients to send total RNA. However, if you need any extraction services, you can always feel free to contact us. After we receive the total RNA, we will move forward to library construction, sequencing, and bioinformatic analysis. Before each step, we will perform quality control to make sure we are taking care of this project very well at each point. On the right, in this pie chart, it is showing the components of the total RNA, and we can see there in the total RNA, there are a lot of ribosome RNAs and a very small proportion of messenger RNA and non-coding RNAs. To target each type of RNA, we propose different libraries, such as for messenger RNA sequencing, as they all have the poly-A tail in those structures. We will use poly-A enrichment to prepare the library. While for other non-coding RNAs, for example, long non-coding RNA, we will use ribosome removal and directional library to make sure we enrich long non-coding RNA significantly and efficiently. We also provide whole transcriptome sequencing service. In this one, you can sequence all 4 types of RNA in a single run. And in this service, we will only need to prepare 2 different libraries. One will be the long non-coding RNA, and the other will be the small RNA library. If you have your own preferred library construction protocol, or you want to send us some pre-made libraries, we will also offer that. And to sequence the messenger RNAs, by using different platforms, we can also perform the isoform sequencing or iso-seq. And in this one, we will offer the PacBio RNA library to perform the RNA sequencing on PacBio platform. Well, in a lot of messenger RNA analysis or research, we will focus on 2 questions. So we would like to know what kind of RNA are there in my transcriptome. That will be the annotation of my transcripts. And we also want to quantify each of my transcript. That will be the quantification of my transcriptome. And here, this pipeline is the messenger RNA analysis pipeline using the NGS data. So we start from raw data and we perform data quality control. And in the two below branches, we will answer those two questions. First, we perform the quantification of our transcripts, as shown on the left branch. That includes the gene expression quantification, differential expression analysis, and functional analysis to see if there is any significant biological process has been enriched in my samples. Well, the second one, as shown on the right, will be the structural analysis that will deliver any mutations identified in my samples, including the SNP or indel mutations. And we also include alternative splicing analysis to see if there is any new transcripts or new isoforms showing up in my transcriptome. For human projects, we also provide other analysis, including fusion gene analysis, oncogene annotation, reactome enrichment, and DO enrichment. As I mentioned, so that pipeline will be used for the NGS data. However, by using the short reads, there may be some disadvantages when we analyze the RNA-seq data, such as they cannot cover the full length of my transcripts, and sometimes I may need to assemble them, so that will introduce some bias or even errors. Well, the other option, the isoform sequencing using PacBio, will avoid those kind of issues or deal with those challenges very well. So as shown in this slide, by using the isoform sequencing, it will cover the full length of the transcripts, and there will be no PCR bias by using PacBio sequencing method. However, by using this platform, it also has some disadvantages, such as it will have a little bit higher error rates, and you need to prepare very good quality RNA as the input materials. And by using the PacBio data only, you cannot perform the quantification as the NGS data do. So that will be the everything for the coding RNA or the messenger RNA. Well, for the non-coding RNA, in addition to the annotation and quantification of my transcripts, we also want to check their potential targets. As shown in the figure, a lot of RNAs can either interact or compete with each other or different types of RNAs to involve in the gene regulation. So that's why in the analysis part, we also include the target prediction in our package. Here, I listed the long non-coding RNA-seq pipeline as an example. So we include the long non-coding RNA identification and the target prediction in this analysis pipeline. By including this analysis, we will be able to profile not only transcript abundance and novel transcripts or genes, we can also discover the variants and predict the target genes, and even explore the regulatory networks by studying them. So that will be used for the individual non-coding RNA-seq. Sometimes we would like to see the global map or the interaction or the even network between multiple types of RNAs. To achieve that research objective, we propose the whole transcriptome sequencing service. In this one, you do not need to sequence each type of RNA individually or separately. You only need to send us, again, total RNA, roughly 5 micrograms, and we will prepare 2 different libraries. One will be the ribosome removal and directional RNA library to target long non-coding RNA, messenger RNA, and circular RNA, while the other library will be the small RNA library to target the small RNAs. And in the analysis part, we will provide not only the analysis for individual RNA, but also the free joint analysis to study the network between two of those types of RNAs, or even among three types of RNAs, as shown here. And if you compare the input requirement in our whole transcriptome sequencing versus the individual service, you don't need to prepare that much, roughly 10 micrograms to sequence all of them. You only need half of them. 5 micrograms will be enough for us to perform this. So that will be everything for the RNA-seq. And for the epigenome sequencing, we will provide you the whole genome bisulfite sequencing to sequence the methylation level, or even RIP-seq or ChIP-seq to study the interactions between the micromolecules. So let's check them one by one. For the whole genome bisulfite sequencing, in the traditional way, we use bisulfite conversion to convert unmethylated C to T for sequencing, while the methylated C will remain as C in the sequencing part. However, by using this traditional pipeline, including DNA fragmentation, followed by adapter ligation and bisulfite conversion, in this step, the conversion introduced a lot of DNA damage, and with that, we cannot use the input library as much as possible because they break them down into small pieces. So with that, we do need to prepare a lot of input. However, this will be a limit for some very precious materials. So we propose the low input method or the post-bisulfite adapter tagging method. In this one, followed by the DNA fragmentation, we perform the bisulfite conversion first to transfer the unmethylated C to T, to transfer the unmethylated C to U. While follow that, we will perform the adapter ligation. And with that, even though they introduce a DNA damage, but we still can ligate the adapters to both sides. With that, we will use the input as much as possible. If you compare the sample requirements between those two methods, in the traditional method, you need to prepare roughly 2.5 micrograms of your DNA. However, by using our method, you only need to send us 100 nanogram or even 20 nanogram for the cell-free DNA. So that will be enough. That will be enough for us to perform the whole genome bisulfite sequencing. And in the analysis part, by studying the data, we will find out where the methylation is, and we will also quantify the methylation level for different scales. And by comparing different samples or groups, we will be able to find out if there is any differential methylation in different samples. For the epi-seq, we will use ChIP-seq to study the transcription factor targeting region or histone modification. And we suggest the RIP-seq to study RNA stability and structure and microRNA targeting region. So in those epi-seq technique, DNA or RNA will bind with protein of interest, and then we will use the antibody to capture those complexes. After they release the DNA or RNA, we can perform the sequencing. And by studying the data, by looking into the data, we will find out where the enrichment or where the peaks are across my whole genome. So through peak calling, we will find out which region in those genomes will bind with this protein of interest. And within those regions, there may be some conservative sequence as shown in here. So we'll perform the motif prediction to help you identify the conservative region to achieve those interactions between protein and DNA. And we can also compare between samples or even groups to see if there is any differences within peaks across samples or groups. So with that, we go back to this service map, and actually more and more researchers are using multi-omics to perform to achieve their research objectives. If you are interested in those discussion topics, please feel free to join us on day 2, and we will have the mastermind session to discuss the future of multi-omics in scientific research and clinical diagnosis. With that, I'd like to thank you all for your attention, and if you need any help or if you are interested in our service, please feel free to contact our local representatives. And thank you again. Have a good day.